I'm Chad Robbins, the CEO of Adaptive Biotechnologies in Seattle, Washington. Adaptive has developed an assay to characterize the adaptive immune repertoire. Specifically, we're able to sequence T-cell and B-cell receptors at a level of depth and specificity that was previously impossible without the advent of next-generation sequencing technologies. First, we developed a research-based assay to get this technology into the hands of researchers to be able to analyze the work they're doing across therapeutic areas. Specific to this conference, we have an assay that we developed for monitoring minimal residual disease in hematological malignancies. How this technology works is we're able to first identify lymphoproliferative clone and tag it. And then after the patient undergoes a myeloablative procedure to try to knock down the clone, we can then identify at a very accurate and sensitive level the pre-identified clone, with the hope being that we're able to predict relapse earlier for more accurate treatment decisions for the patient. We also do a significant amount of work in cancer immunotherapy, both in analyzing predictive response to the checkpoint blockade and in adoptive T-cell transfer. CAR T-cells, one of the applications is really just an extension of monitoring minimal residual disease, is to determine whether the therapy worked or not. Secondly, within solid tumors, we do a significant amount of work in the checkpoint inhibitors in four different areas. One is to characterize and help identify mechanism of action. The second is to identify predictive biomarkers of response. The third is to look at adverse events. And the fourth is to look at combination therapies and sequential therapies. These are exciting areas of research that we're doing kind of multiple studies across tumor types on to be able to really develop an assay for a predictive kind of biomarker response to really what is one of the most exciting new areas in the drug world, which is the checkpoint inhibitors.